we are really focused on the interactions between skeletal muscle and the extracellular matrix. So we came up with the term myomatrix lab for the lab, combining the myo and the matrix. And there's really two sort of main arms to our research. One is using some advanced imaging modalities supplemented by biochemistry to really understand the architecture of the muscle and particularly its extracellular matrix. And then we also want to know how that relates to the mechanics. So in patients with neuromuscular diseases, the formation of contractures where the muscle becomes excessively stiff are quite common. We want to know what structures are really responsible for that stiffness. And then alternatively, healthy muscle is really quite good at adapting to meet its demands, but that is compromised in these neuromuscular diseases. So we want to understand how that altered architecture impacts that adaptability, and particularly the response of the muscle stem cells that are present in that environment. So how can that altered matrix really lead to the mechanical dysfunction as well as the impairment in regeneration, so that we can develop novel techniques and therapies to target those specific structures or architectural features to enhance both the function and regenerative capacity of muscle in neuromuscular diseases. So one of the things that we'd really like to make progress in is transitioning from some mouse models into more human neuromuscular diseases. So in the case of cerebral palsy, the development of these contractures are really quite common, unfortunately, but there's really no good animal model. So we are starting collaborations with orthopedic surgeons to access biopsy tissue during surgeries, so that we can take these muscles to the lab and really probe them and understand what are the architectural features that create such a stiff muscle in contracture. And then we also want to expand our knowledge of the stem cells that are present in muscle and how they respond to the matrix. So beyond just the muscle progenitors themselves, but the stem cells that influence them, and particularly the mesenchymal stem cells or fibrodipogenic progenitors, how they adapt to that altered extracellular matrix environment in fibrosis. My favorite thing about ORS is that it brings together a lot of people who are interested in musculoskeletal research from both the basic science side as well as the more clinical aspects that are, I think, really important for translating a lot of our work into the clinic, so that we can foster these collaborations with our orthopedic partners to access these human muscle tissues and really drive the research forward in a way that is translatable and important for actual human health, not just that of the mice.